Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension. And on behalf of those folks and our other co-organizers, Wisconsin Public Television, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to introduce to you Elle Kielar-Grevstad. She's in the Department of Biochemistry. She was born in Woodstock, Illinois, and went to high school in McHenry, Illinois. And then she came up to UW-La Crosse to double major in both biochemistry and in cellular and molecular biology. Then she came to UW-Madison to get her PhD in biochemistry. And then in 2014, she returned to UW-Madison to help launch the new Biochemistry Optical Core. Tonight, she's going to talk to us about from cheddar cheese to single synapses, light microscopy across the disciplines. And I must say there are very few scientific words that rhyme with cheese the way that synapses do. Way to go. This is going to be quite the visual treat. Things have changed a lot since I first put eyeballs to a microscope back in about 1971. First of all, the world is in color. Second of all, you can record it. Third of all, it moves. And fourth of all, things can be alive. And that's pretty astonishing things. It's great to see where microscopy has gone in the last 30 years or so. So please join me in welcoming Elle Kielar-Grevstad to Wednesday Night at the Lab.
Can everybody hear me okay? Pretty good? Okay. Thank you all for having me. Thank you, Tom, for the invitation to talk. We spoke in the hallway one time, and I had talked to him about about our core facility and how we get people who image cheese, and then in the same day we have people who are looking at neuronal synapses. And he thought that was just fantastic, and he wanted me to speak with you guys about the breadth of research that microscopy supports on the UW campus. So the title of my talk today, like I said, is From Cheese to Single Synapses, Microscopy across the disciplines. I'm headquartered just across Henry Mall in the biochemistry department, and I'm the director of the Biochemistry Core Facilities. Just to give you guys a sort of perspective on what the Biochemistry Core Facilities mean and what it means to be a core facility is you can think of us as sort of a community lab space. People from all across the university as well as outside of the university come and access the expertise and the instrumentation that's in our core facility in order to do research. There are 4 core facilities on campus, so I've just highlighted this. This is us in red, the biochemistry department. We're here tonight, right? So there's 4 other light microscopy core facilities on campus. The one that I'm highlighting is the Biochemistry Optical Core, and then there's also one over the Wiseman Center that supports brain research. There are a couple over at the hospital in the Wimmer. And then there's also one in plant in the botany department that supports plant research. One nice thing about the Biochemistry Optical Core is that we're sort of centrally located. So we get lots of different types of researchers that come to our campus and come to the core facility to use us. So just for instance, right here, if I can find my cursor, biochemistry is here, genetics is here, metamicro is here, horticulture is here, food science is here. So all of these people come and use us, as well as the engineers across the Great Divide of Campus Boulevard. So in 2014, I was asked to come back to the university to start up the Biochemistry Optical Core. And our explicit mission is to provide support and train the research community on the techniques and equipment that encompass basic to state-of-the-art light microscopy methods. We were formally founded, like I said, in the spring of 2014, and since then we have over 200 different researchers that we've interacted with, and those are from about 80 different research groups across 27 different departments on campus. And that represents about 7 different colleges and schools. You may be thinking, oh, how does a food scientist use a microscope in the same way that somebody from, say, psychology would use the microscope? And the answer is that they don't use a microscope in the same way. But they're all interested in the spatial orientation of molecules or proteins or structures in an environment. And just to give you guys a background, I think the easiest way to wrap your head about how these multiple disciplines can leverage microscopy, I want to sort of bring us back in time, all the way about 350 years ago, and start talking about how the sort of invention of microscopy and how the principles of that invention still remain the same today, as well as what has pushed it forward to make it more of a quantitative and dynamic field than it was perhaps in the early 20th century. So the father of microscopy, his name was Anton Van Leeuwenhoek. He developed these simple microscopes, and for those of you who are in the audience, what you were looking at when you came in are images taken on replica microscopes. So those are, what they did is they put pond water on this specimen holder, held the microscope up to a candle, and looked at the tiny organisms within that. What you would see, right, if you were that person, you would see something like this. And immediately, as a scientist, and as my 5-year-old daughter and my 2.5-year-old son said when looking at this, they said, "Mom, what is that? Why are those things so small? That's a whole different world. I didn't know there were little things that grew in water." Well, and my daughter's quite bright, and so she was like, "Well, why is that thing crawling on that? How does it know to crawl on it, right? What are those bright dots? Why are those organized this way? Oh, what is that? Why is it slithering through the space and it's not interacting with this?" So you can imagine the sort of vast explosion of this small, tiny world that Van Leeuwenhoek discovered, essentially. And you can see how many questions you can ask just by seeing something. This continued, so microscopy continued to be sort of a fad for a long period of time, and this is an image, if I can stop this, which would be helpful, and move forward. So this is an image in the 1800s, the Victorians loved microscopes, right? In 1846, there's these wonderful quotes by the naturalist Charles Kingsley, who explained the tiniest pieces of mold on a decayed fruit and the tiniest animalcule, which is a tiny animal, from the stagnant pond will imagination find inexhaustible wonders and a fancy of fairyland. And then in 1861, before he became the president of the Royal Microscopy Society, Henry Slack described a group of protozoas as a tree from fairyland, right? We laugh, we giggle, like fairyland, that's so crazy, right? Why would they call them fairies? But if you think about this is 200 years ago. I sort of chuckle to myself and I think, well, 200 years from now, right, we're going to be in the 23rd century, hopefully. What are the top scientists in our field going to giggle about that we thought we understood very, very well? Probably a lot, I'm assuming. But why is microscopy in the 1600s and the 1800s? What's this thread? What is the connectivity, and how are the microscopes of today, what do they have in common with the microscopes of 350 years ago? And the answer is actually quite a lot. So A modern microscope would look something like this. You would have a computer attached to it, so you could digitize your images, and so you wouldn't have to be an amazing artist to be able to draw the fine synapses or structures of cheese that you were looking at. But they all have three common things, is that they magnify an image, so they make it bigger. They magnify an object. They make it bigger so that your eye can see it. They resolve details, which is different than magnification. So resolving details means that they can separate two objects in space from each other. And you can see those two objects as two distinct things rather than one blob of a thing. And the third thing that they have in common is they send the images to either your eye or to a detector. And these detectors can be quite simple, like your iPhone, or they can be as fancy as a $100,000 camera. So there's a vast variety of ways in which you can capture images on a microscope. But they all have one fatal flaw, like microscopy does. And it maybe isn't fatal because the field is still alive and going. But they have a limitation, and that limitation is light. And it is the resolving power of light, which is what we term in our field as the diffraction limit. So if you are going to come to a microscope today, you would want, and you had an organism, anything from a mouse to a small molecule that you wanted to study, you would have to pick the type of microscope or the type of imaging station to fit your needs. So anything from a mouse you could image with a PET or a MRI or an ultrasound. You could even use a confocal in some unique settings. If you were trying to image mouse brains or maybe an ant or a hair, which is about as fine as your eye can see, you could bring it to a standard microscope. Once you want to start imaging single cells or bacteria, they're still suitable to see with a microscope because they're right around 1 micron in size. But once you get below 200 nanometers, which is the diffraction limit of light, you're no longer able to resolve individual structures from each other. So say you had 2 viruses that are packed within a cell, and they're close to each other, you couldn't tell whether or not those are 2 viruses or 1 virus, right? And if you were looking at the structure of proteins, which are on the order of 10 nanometers, you couldn't use light microscopy to see those individual molecules unless you did a sophisticated form of microscopy. But what you can do is you can use microscopy in order to give you spatial relationships between the organizations of proteins within cells, or the organization of viral particles within an organism, or how a bacteria moves within space. And the reason why the diffraction limit is, so I say it's the diffraction limit of light, and I just sort of want to explain to you why it's 200 nanometers. Because I think even in graduate school, I never really, that was thrown out there. It was said, oh, it's the diffraction limit of light, it just is what it is. And I never really understood it until I had to start teaching it. Like a lot of things that happen in life, right? So, so say if you want to think about why light microscopy is limited, you have to think about like what does a lens do? And the lens, the thing that sees the object that you're looking at, which is the most important part of a microscope, and it is the reason why Anton Van Leeuwenhoek is considered the father of microscopy, because he was such an artist with his milling of a lens that it allowed him to see these small animalcules that he would see. You have to think about, okay, we have one point, and this point is emitting light in some way, right? It's releasing light. And you're using a lens, and this is a modern lens that has some writing that means something to microscopists on it. And what does the lens actually do? So the lens is actually going to pick up the light that's coming off of that object, and it's going to try to reconstruct the light in a particular manner. And lenses are actually not that great at doing this, surprisingly, because light acts as not only a particle, but a wave, right? And so when it comes in, it's going to bounce off the pieces of glass in the lens and interfere with each other. So what you'll see when you're focusing on a single point, and you focus up and down, is that you'll actually start noticing these patterns that arise. So that's a single point, and then, right, this is a sphere, and it shouldn't start looking like a donut over time. I can do that. Technical difficulties. I'm not a sound person, right? We use light. So just to pick up where it is that this single point of light is, if you think of it as a ball, if you focus up and down on it, it shouldn't ring out like this. It should stay as a single object. But because light interferes with itself in odd ways, because it's a particle and a wave, when you project what you're seeing, what you actually see is something that looks like this, which is what we call a point spread function. So the point is here, and then the spread is what you see when you focus up and down. It's also known as airy disks, after a scientist who described the mathematics around this. And this is a problem, because if this single point is 2 nanometers, right? This single point of light, it's coming off an object that's 2 nanometers in size. The point spread function essentially spreads the light at anything greater than 200 nanometers, which is 100 times larger than what the object actually is. And if these 2 points were very close to each other, you wouldn't be able to tell one point from another. And I think that this video that I'm going to show now sort of shows how and why waves interfere with each other. So if you look at this, if you think of those 2 balls as a single point of light, or 2 single points of light, you can see the patterns right here. And then as you come out, it just looks like 1. Does that make sense? So you have 2, and then they converge, and they do they undergo constructive and destructive interference, and it becomes a singular wave. And if we sort of make a cartoon version of this, what you can see is there's 2 molecules in space, and they're both going to emit light. And what happens is that these areas in which these circular waves overlap, it gets amplified over the background. And if you have a lens in its place, what it's going to detect is the bright parts. And so you get this, and so it ends up looking like a single molecule in between these 2 objects. And this is what we call the diffracted orders of light. So this is 0, -1, -2. And for those of you guys who are really interested in light, you need at least to capture one diffraction order off of the 0 in order to resolve objects. So if you think about this a little bit closely, if you have a lens, people will say, well, why don't you just make a lens that's giant and it would capture all of those things, right? Well, that you could, but not really, because there's physics involved, and manufacturing technology is not quite there to make a giant lens that could fit to see a 2 nanometer object. But you can play along with a little bit. So for an objective that is small, you would see this object like this. It would show up as a singular blob. If you had a larger objective, something that could collect here and here, which we have, you're now able to start separating these objects in space. What happens, though, is when these objects become closer together, you can see that these overlapping patterns actually become tighter, right? And so what happens actually is that it's tighter but also spread out for the single order of light, so the -1 and +1. And so you'll see that here. And so even with the large objective, you're still not going to be able to capture the -1, and they blur together. So that's the diffraction. That's how I like to think about it, the diffraction limit of light, and that's the limitation of microscopy. So even though these limits have always been in place and have been known about for a long period of time, since the early 1900s and even into the 1800s, what has changed, right? Why is light microscopy nowadays so much more powerful than the light microscopy that was performed in the 70s when Tom was in school? They still magnify and they still resolve details and they still send the images to the eye and the detector. But what Van Leeuwenhoek would be able to see, which is something like this, and this gets a little muddled. This is a C. elegans, which is a worm that's used for developmental biology to study development. You can see its little heart pumping here. But what you notice is that this would be taken on like a Victorian microscope. And what you can see nowadays is something that's much different. And this is the same organism. Wait. Well, these things move around in space. Maybe we can get it to go, if I can go like this. So you can see them sort of move in space and then with real time. There we go. Technical difficulties. Yeah, it's called GLONASS, which is kind of funny. So this is now CRISPR engineered C. elegans to highlight the gonads of the C. elegans. And so the difference here was the development of fluorescence. And what is fluorescence, right? It's the thing that provides contrast and specificity in microscopy. And for those of you guys who aren't familiar with fluorescence, I'm just going to briefly cover it. So it's the game changer. It essentially means is that you can have a molecule which is dark, which does not glow at all. It's not like a firefly in that regards where it's constantly emitting light. But you can hit this molecule with a certain wavelength of light. In this case, it would be green light. And then that molecule takes up that energy from the green light and does some conformational change in its energy bonds, and it will release the energy as light at a farther wavelength. Usually it's red. So that's just sort of depicted here. And you can see that also in a pretty basic, you can do this at home. My kids got a kick out of it when I did it. So you can take a UV light source and put some tonic water, if you have the real tonic water with quinine in it, and you can shine light into it, and the quinine absorbs the UV light and emits blue fluorescence. And it's the same thing, but it's clear, right? And what is fantastic about the fluorescence is that as soon as you shine light on it, it becomes, it gives you contrast over a background. And fluorescence was actually discovered, not surprisingly, during the Victorian era, in the 1800s. It was, these are just sort of some molecules. They're on a whole order. A couple of these molecules, fluorescein, which is still common today, was synthesized in 1871. And these are all derivatives off of quinine, most of them. So these are chemicals that are fluorescents, but there's lots of different ways. There's lots of different other molecules that are also fluorescent. And then and the chemists in the last 15 and 20 years have been incredibly creative in ways in which to attach fluorescent moieties to proteins, to lipids, to anything under the sun that you want to study. You can make it fluorescent, and you can make it any color that you want to see, which means that you can see multiple colors simultaneously and get a relative organization of up to 20 different proteins or 20 different molecules at the same time. The other thing that was quite revolutionary in the fluorescent field was the discovery of GFP. So for those of you guys who don't know, GFP is taken from, it's called green fluorescent protein. It was first discovered in the 80s and cloned out of that organism, which is jellyfish. It's a jellyfish protein. And then Roger Chen, who won the Nobel Prize a few years ago for the development of all the different colors, he was able to change, just by changing the amino acid structure within the protein itself, change the spectrum in which it gets excited and gets emitted by. And then, not surprisingly, biologists were clever again, and CRISPR came along, and now you can genetically engineer anything you want, essentially, and make that protein fluorescent at endogenous levels in an organism that you're interested in studying. So what did it, I've hinted at this a couple of times, so what does it really provide us? Fluorescence provides us a form of, so you can see this is a sort of flat image taken with a bright field microscope, so similar to what you would, with a modern version of Van Leeuwenhoek's little candle. It provides you contrast and the ability to target the proteins that you're interested in. So this is the same neuron, and now you can see the dendrites and the spines. You can see the sort of network of, I think this is actin within here. You can see the mitochondria, and you can see the nucleus, all with different colors. You couldn't see that sort of nice signal to noise over the background with just a flat image. So this was revolutionary, right? And has been. And so how are people on campus now using these sort of revolutions, right? It's not just to take pretty pictures. This is one of my pictures. For those of you guys who don't know, I love taking, using art or using microscopes and promoting art through it. So this is an image of a piece of corn. It's a seed. It's a cross section. This is the embryo of the corn. So this is the sheath, and then this is the endosperm. This is taken on a microscope that allows you to take optical sections of this image, and it's fully spectral, which means that I can take any color on the visible light range and see it. This is great, but and it's beautiful work, but it doesn't really address scientific questions. It just sort of gets people interested in science. So this is maybe less interesting, but maybe a more research, maybe it's not less interesting at all. John would be very upset at me for saying that. This is maybe less detailed or less beautiful in terms of structure, but John Lucy, who is the director of the Center for Dairy Research, his group brings mozzarella to us, and they'll take a chunk of cheese and they'll slice it on our wet bench, and they'll stain it to understand the protein structure of this cheese. And what you're seeing here is cheese that is a week old, cheese that is 3 weeks old, and cheese that is 6 months old. And what you may be able to tell is that the structure changes over time. And this is actually a breakdown product. And so John's really interested in trying to create manufacturing processes and natural derivatives in order to stabilize the cheese structure over time. And he uses the microscope in order to see that structure and to see whether or not his experiments are being successful and whether or not he can apply the work that he's doing to the dairy industry. In another applied research setting, this is, I wish this was a little bit, this is one of my favorite images only because people get so excited when I tell them what it is. So this is out of Daniel Naguara's lab in civil environmental engineering. Daniel's extremely interested in trying to engineer wastewater treatment plants to be more efficient. One thing that is one of the most costly things in wastewater treatment is the oxygenation of water, so that the microbes that scavenge the nutrients out of the water can work effectively. He's trying to engineer bacteria that function under low oxygen conditions. And so he can look at the RNA that's getting produced in the wastewater sludge. So this is sludge from a reactor in the wastewater treatment plant. It's essentially a biofilm, so it's all of the organisms that make up this sludge, and he's staining for the different genetic factors that could promote more efficient clearance of phosphorus and nitrogen out of the wastewater. It's pretty amazing that this is sludge and it's so beautiful. I think it, personally. I was really excited to see this. So if you start thinking about this, it's like these are tiny organisms, right? So you may be thinking, oh, well, is this really relevant to into the medical world? And I would say yes, because of people like Dr. Bruce Klein, who's in medical and microbiology and immunology. And for those of you guys who can't see, this scale bar is actually down here, right here, is actually 1,000 microns, so 1 millimeter in size. This is a live rat lung, or mouse lung, sorry. So it's a cross section. So it's huge. This is a piece of tissue that you can see with your eye. What we're seeing is all the individual cells, and these little tiny red dots are fungus. So Dr. Klein is especially interested in studying how fungus induces asthma in people and how your immune cells respond to a pathogen within your lung. And by understanding that more by the basic understanding of how these things get arranged, and these green guys are actually what are called these guys right here. These are immune cells that have infiltrated the lung and become activated in the presence of fungus. And he is trying to understand how the spatial orientation of the fungal infection activates and exacerbates an asthmatic attack. So this is live tissue taken over about 30 minutes in time, and we were able to see that the spatial orientation is in fact important for the activation of these immune cells. This isn't so dynamic though. It's not like a movie. We can't watch it in person. We can't really see the immune cells coming in and out. But if you wanted to start doing that, you can do that rapidly on a new microscope or a modern microscope. So this is taken out of Melissa Harrison's lab. She's in biomolecular chemistry, and she's extremely interested in how maternal factors drive development. So maternal factors are inherited by the mother, and they are incredibly important in the first steps of development in an embryo. So this is in Drosophila. These are CRISPR-engineered organisms. And what you're going to see is the red you're going to see single transcripts get transcribed, and that's the green. So that's DNA getting transcribed in a living organism in real time. The red, for those of you guys can see, mark the histones of the DNA. We can push this a little bit farther, and we can push this into a more of an adult Drosophila that's further along in the development. And Dr. Hong Zhang, and he's in genetics. Sorry, is explicitly interested in studying the organization of how a neuron comes onto a muscle and causes contraction. And is also interested in these sort of what they're called neuromuscular junctions, where neurotransmitters are released into space. And he marks these neurotransmitters with by calcium channel, and you can multiplex this over time. And you can watch the synaptic these junctions be activated and release neurotransmitter in real time. So this movie is going to be taken from Dr. Kate O'Connor-Giles' lab. She was in genetics and had moved to Brown last summer. And what you'll see is that, so that's the same junction, it's just zoomed in now. And the spikes are actually single synapses being released. And she's incredibly interested in understanding the role of the synaptic heterogeneity, so the probability that a single synapse will release, and how that develops neuronal circuits that are important for learning or important for anxiety-related disorders. So she was able to map each individual active zone with events in real time, and then look at the probability of release, and then correlate this to behavior. If you want to study this, maybe we had mentioned the word vesicles earlier. I don't think it got captured by the broadcast. But in my own work, we were studying a vesicular trafficking at the plasma membrane, so the release of hormones into the extracellular space. You can think about it, insulin being released out of a cell, and how that gets regulated by the protein environment around it. So if we go to the next slide, each one of these green markers are a vesicle, a small packet of hormone, and when they get bright, it means that the vesicle is opening into extracellular space and the content is being released. You can simultaneously image at the same time another channel. So in this case, this is the cytoskeleton of the cell. And you can watch these at the same time and understand the dynamic behavior between one protein and another protein. But there's a catch, right? Like, this is super, the red on here is pretty diffuse looking, right? And that's because the diffraction limit of light is not allowing us to see these fine structures. So I think it was in 2014, the Nobel Prize was won by Stefan Hell, William Moner, and Eric Betzek for the development of super resolution microscopy. And that breaks that spatial diffraction limit of light. And so during that time, we had known about this is prior to the work being done, or the Nobel Prize being awarded. Here at the Optical Core, we had developed a technique based on their technique in order to study the cytoskeleton of these cells in greater detail. And so what you do under this is that you take a movie very, very fast. So this is 100 frames per second. And it's hard to see, but there's individual blinking events. And what you can do is that's a single molecule blinking in time. And you can use math to essentially plot that molecule in space and fit it with a mathematical function that allows you to estimate where that molecule is in space with higher resolution than what the diffraction limit gives you. And so if you do that, and you add up, it's essentially pointillism. I like to think it like that. So if you do that, over time you can start seeing, this is the same image, and you can start zooming in, and you see these really fine structures which are starting to get blurred out because this camera has pixels and is binning the information. And you can zoom in even more. And what we know is that the individual vesicles lie within these spaces, right here, and that over time, if you trigger the cell to release its hormones, this network, the big fibers get cut and they get dissolved, and then they come back. And that's very important for the regulation of the amount of neurotransmitter that's released from each individual vesicle and then total number of vesicles that are released over time. So with that, I just sort of want to end with one of my favorite quotes. So I have spent more time than many will believe making microscopic observations, but I've done them with joy, and I've taken no notice to those who have said, why take so much trouble and what good is it? That was taken from Anton Van Leeuwenhoek, and I still think it applies to me today, 350 years later. And with that, thank you for your attention. Are there any questions? Are you taking any questions? Yeah.
Do you think there's any limitation with fluorescence imagery and digital microscopy when utilizing the XYZ axis for formation of cells?
I'm just going to repeat your question so that everybody can hear it, and for me to understand it. Do I think that there's any limitation for fluorescent microscopy in not only the XY plane, but in the XZ plane?
XYZ.
XYZ,
yeah.
The
so
whole 3D.
A composite picture
yeah.
at the same time.
So there's limitations, especially in standard microscopy. You can get three-dimensional images, and that's not difficult to do. But the stretch in the Z dimension, so we talk about the diffraction limit of light, and typically the numbers are thrown out for the XY direction. So how well you can, on a two-dimensional plane, how well you can separate one molecule from the other. That's 220 nanometers typically. On the Z plane, it is double that. So if a molecule is up here versus down here, they have to be about at least 600 nanometers apart to tell whether or not they're one object or two. And that's just how the stretch of light, how it works with the stretch of light, because it's not symmetric. So to do the 3D volume rendering, what you do is that you can basically now take the state, you can take the lens and you can move it up through the focal plane. And there are computational tools that allow you to then reconstruct that image and rotate it in space and see it in 3 dimension. And that's actually very standard form. The person that developed that sort of computational method and the technique, his name is John White, and some of you guys might know his name. He's a very famous microscopist. He developed the first confocal microscope, or commercializable form of a confocal microscope. He did that work in Cambridge, but in the early 90s came here and was a professor of genetics or molecular biology. In his seminal work, he basically took a C. elegans and mapped all of the neurons in the C. elegans over time. So he took slices of the C. elegans, and that work was done in the mid-70s. And he sort of developed what is now termed of he's like the father of connectomes. So you could see this entire neuronal circuit in this transparent molecule, in this transparent animal. Does that answer your question?
Yes.
Okay. Yes, Tom.
What would you be able to tell the folks here about the cryo-EM initiative?
Right. So Tom asked a question about cryo-EM microscopy. So EM stands for electron microscopy. It's different than light. Instead of shooting photons at a sample, you're shooting electrons. There has been a big revolution in the field of electron microscopy with a form of electron microscopy called cryo-EM, which allows you to now start seeing structures of proteins. So you're getting the same sort of like resolution in XY space as on an angstrom level. So instead of at a nanometer level, you're now 1 or 2 orders of magnitude lower than that. The equipment that is necessary and needed for that type of imaging is incredibly, as you could imagine, expensive and sensitive, and the biochemistry department just recently invested in that technology, and we're currently under construction for the Center for Cryo-Electron Microscopy at UW-Madison. So hopefully by the end of 2019, it will be, the instruments will be installed, and you'll be starting to be able to see dynamic protein changes, which is a real barrier when you're looking at structural biology. So when you're looking at protein structures or structures at that level, one of the big barriers right now is that you have to sort of freeze the molecule, and you can't see all of it, how it breathes and how it moves in space, which is incredibly important to its biology. Does that answer your question?
Yeah, now how do you do electron microscopy with living stuff?
You don't. Because you're shooting, well, on the Krios, so this is the name brand of this microscope that's coming, it's called the Titan Krios. It's frozen in, this was just cryo part, it's frozen in vitreous ice, which has no crystals in it. So it's frozen very, very quickly, so you can see through it. So the things don't survive that freezing step. And then you're also shooting 300 kilovolts of electricity at it, so it burns them pretty fast.
So you like microscopists still have that advantage over other folks?
For the meantime, maybe in 200 years, we'll be laughed at. Is there any other
questions?
Any other? Yes.
I'm assuming that the researchers that come to you from the various departments around campus probably have to pay fees for the services you guys provide.
Mm-hmm.
Do firms from the outside of the university, so for example, some of the private or commercial firms up at the Science Park along the west side, do any of those firms who are doing a lot of research, drugs and various things,
They
come
do.
to you guys and pay you for some of these services, or is it just university?
So the question for those, just to reiterate, is that are our clients solely academics from the university, or do we get industrial users from the biotech community in Madison? And the answer is yes. We do get industrial users for the biotech community center. They do not pay an academic rate. They pay an industrial rate. So it's more costly for them to use our facility, but it's much cheaper for them to access us than for them to buy their own microscope at the same time. Does that answer your question? Mm-hmm. Yeah. Any other
questions? If not, thank you very much.